Hello everyone. Today we are going to discuss about connective tissue and its staining. We'll start with the various methods used for studying connective tissue through the histopathological procedures. Following are the staining methods used in the histopathology lab so that we can study the various connective tissues. First one is a trichrome staining. Second is a Verhoeff stain. Third one Weigert Resorcin stain. Fourth one Gordon's and Sweet stain. Fifth one Gomorrah stain. Sixth one is a von Giosen stain, and the last one is seventh is the PTS stain. In this slide we will be knowing about the various types of connective tissue. There are three types of connective tissues. First one is a loose connective tissues. Second is a dense connective tissue, and third one is a specialized connective tissues. So loose connective tissues subtypes are areolar, adipose, and reticular. Dense connective tissue subtypes are dense, regular, dense, irregular, and elastic. And the specialized connective tissue examples are cartilage, bone, and blood. Now, while studying connective tissue, we should know what is collagen. The most abundant protein in mammals. This collagen is the most abundant type of protein in all type of mammals. This is basically the main component of the connective tissues, and 25 to 35% of the whole body protein is collagen. In collagen, the major amino acids are like proline, glycine, and hydroxyproline. These are the subtypes of loose connective tissues. First one is areolar. This is basically found between the different organs and connect the skin to the underlying muscles. This is also seen in the skin and muscles and in the bone marrow, as well as around the blood vessels and nerves. Basically blood vessels and nerve, wherever it is found, so surrounding this blood vessels and nerve, these areolar tissues are found. So next one is a adipose tissue. This is basically found under the skin or between internal organs. Sometimes they are also found in the inner cavities of the bone. That is basically the marrow adipose tissue. And the third one is the reticular tissues, where this type of tissues are found in the endocrine glands, liver, bone marrow, and lymphoid organs. This is the second type of connective tissue, that is called dense connective tissues. And here in this slide, there are two dense connective tissues are shown. One is a regular and another is a irregular. So dense irregular connective tissues are found in the ligaments and the tendons. And dense irregular tissues are found widely throughout the body. They are widely distributed throughout the body. For example, pericardium, endosteum, periosteum, heart valves, joint capsules, deep region of the dermis and of the skin. This is also one of the type of the dense connective tissues, that is elastic fiber. And they are found in skin, lungs, arteries, veins, elastic cartilage, periodontal ligament, and fetal tissues. Now we'll start with the staining procedures. The first one is a trichrome stain. Trichrome staining is used to visualize connective tissues, particularly the collagen, in tissue sections. After staining with Masson's trichrome procedures, following observations are seen. Collagen will appear blue, nuclei dark brown, muscle tissue will appear red, and cytoplasm pink. This Masson trichrome stain shows three colors in the procedure, and this is the most common staining used in the histology for observing the basically the connective tissues. In this image, you can see the liver tissues after the cirrhosis. Trichrome staining, basically the Masson's trichrome staining is used to study the various disorders like muscular pathologies, cardiac pathologies, hepatic pathologies, kidney pathologies, and hepatic and kidney tumors. What are the procedures? So here, first of all, sample is immersed into the Weigert iron hematoxylin, and there are three different solutions labeled as A, B, and C. Here, Weigert hematoxylin consists of three solutions: ferric iron chloride in diluted hydrochloric acid, hematoxylin in 95% ethanol, and potassium ferricyanide solution alkalized by sodium borate. This stain usually basically color the nuclear part of the cells and tissues. Now solution A, B, and C. Solution A, also called plasma stain, which consists of acid-fuchsin, xylidine, ponci, glycolic acid, distilled water. Solution B usually consists of phosphomaldic acid in distilled water, and solution C is basically the fiber stain, and it consists of light green SF yellowish or fast green FCF. It is used to stain collagen. So we studied that trichrome staining procedure consists of two different types of ingredients. First one, Weigert hematoxylin, which is a nuclear stain, and another is a three different solution, that is solution A, which is plasma stain, B, which is a phosphotungstic acid, basically phosphomaldic acid in distilled water, and solution C is a fiber stain. It also used to stain collagen also. Next is a Verhoeff stain. This stain is also known as Verhoeff's elastic stain or Verhoeff's Van Giosen stain. This stain was developed by Friedrich Hermann Verhoeff in 1908. The purpose of this Verhoeff stain is to demonstrate normal or pathologic elastic fiber. This stain, basically the clinical use is to study the elastic tissue in blood vessel wall, elastic cartilage, lungs, skin, bladder, and some ligaments. So what is the procedure of Verhoeff stain? So here we can start with the fixatives. Any fixatives, formalin-based fixatives, basically gives better results. So you can use any fixative, but the formalin-based fixative gives better results. Here two steps are given for Verhoeff stain. First one is a initial stage, and the second is the later stage. So in the initial stage, we use Verhoeff stain, which consists of hematoxylin, iron chloride, and the third one is iodine solution. So here iron chloride and iodine acts as a mordant, and as a mordant it aid in the oxidation of hematoxylin to hematine, which is responsible for staining the elastic fiber. Elastin basically poses a strong affinity for the hematoxylin iron complex, and sodium thiosulfate here is used to remove excess iodine. Now if you have the coloration of the tissue sample with the help of iodine, so the extra iodine color can be removed with the help of sodium thiosulfate. Now later stage, the initial stage we can stain with the that we discussed is the Verhoeff stains, and the later stage is a Van Giosen counter stain. Okay, so initial stage is a Verhoeff stain, which consists of hematoxylin, iron and chloride, and iodine solution. So later stage, it is a counter stain, consists of agric acid and acid-fuchsin. So after staining with the initial stage and later stage, the observation will be like elastic fiber and cell nuclei will appear black, collagen fiber will appear red, and other tissue elements including cytoplasm will appear yellow in color. So let us study about the Weigert Resorcin stain. This stain is used to identify elastic fiber. Weigert Resorcin stain is a combination of basically two elements, resorcinol and fuchsin. Counter stain here we use fast red or hematoxylin. Now after staining we can see the following observation. Elastic fiber will appear blue, black or black. Collagen will appear pink to red, nuclei will appear blue to black, and other tissue ligaments will appear black, basically yellow in color. This is the image which we can see in the in this picture. This is blue colored elastic fibers are seen, visibles. Now next is Gordon and Sweet silver stain. This is basically used to demonstrate if there is an abnormal reticular fiber in the tissue sample, and normally this is done for the diagnosis of the disorder like cirrhosis or necrosis of the liver. This reticular stain is often used on liver biopsies, and the reticular fiber have less affinity for silver solution, so the tissue is first oxidized using potassium permanganate, and this is helpful in the staining the silver, basically the helpful in the enhancing the staining with the silver stain. Silver deposit can be initiated on the sensitized side of the fiber. So here reticular fibers are visible in this slide. Now Gomori stain is again another type of connective tissue stain. Here we can stain all the tissues content. Basically this Gomori's name was given by the scientist known George Gomori in 1950. He developed the Gomori's trichrome stain, and this is a one-step method staining technique helpful in the identifying if there is an increase in collagen fiber in the connective tissues. It differentiates between collagen and smooth muscle fibers. So observation after Gomori's stain is your nucleus will appear black, cytoplasm, keratin, muscle fiber will appear red, and muscle, basically the collagen mucus, will appear green or blue. This is the Verhoeff stain. Verhoeff stain consists of mixture of picric acid and acid fuchsin. This is the very simplest method of differential staining of collagen and other connective tissues. This was developed by Ira Verhoeff, an American neuropsychiatrist and pathologist. So we can see in the image Verhoeff stain, smooth muscle fiber yellow and collagen fiber red. The last one is a PTH stain. PTH stands for phosphotungstic acid and hematoxylin. PTH is a mixture of hematoxylin with basically phosphotungstic acid. What is the role of phosphotungstic acid? It binds to tissue protein. Here, what kind of specimen we can use in PTH stain? So formalin based or paraffin embedded or frozen section can be used. PTH stain uses. It demonstrate, basically also demonstrate differentiate also, either it is collagen and muscles. It demonstrates striated muscle fiber and mitochondria and identify contraction bands as seen in contraction band necrosia. So this is the image shown in the tissues taken from the heart attack patient, that myocardial infarction, phosphotungstic acid hematoxylin staining demonstrating the contraction band necrosis in individual who had a myocardial infarction. So this is the observation. We can see muscle will appear like blue black to dark brown. Connective tissue will appear like pale orange pink to brownish red, and fibrin neuroglia will appear blue, very deep blue. Coarse elastic fiber will appear purple. Bone and cartilage will appear yellowish to brownish red color. So here we completed about the connective tissues and its staining methods. Thank you for listening.